Hi everyone, I'm Marion Carme and I work with Eduardo and Marta at INRAE in the Biogeco unit. I'll keep talking about delta trait SDM and particularly how we use them to predict early stages traits in some European oaks. So delta trait SDM are correlative models, as you heard in Marta's explanations. They can be located here in Levin's standard classification, and we build them on R. Here is the general formula of a data trait SDM, how they can be built and adjusted. We want to explain a trait. It can be a germination trait like a rate or time. It can be a fitness-related trait like survival, growth, total biomass, and it can be phenological trait, etc. And this trait depends on the population climate, the climate of origin here in blue, that had shaped long-term adaptation, mother tree traits, and seed development. It also depends on its current climate. Here it's the climatic transfer distance in orange. It's the difference between this current climate and the climate of origin. It's basically how plastic this trait is. And this plasticity can depend on the population climate, so there can be an interaction between the two. Here it's in purple. And finally, as Marta said, other traits can influence this trait. So we can combine their model with this model to better predict our trait of interest. So you can already see which kind of data we need. Now let's see precisely which data we collected for modeling early stages of oaks. You can see here that we have 3 oak species covering a gradient from temperate climate to Mediterranean climate. We have sessile oak, Quercus petrea, which is temperate, Quercus pubescens, pubescent oak, that is sub-Mediterranean, and Quercus suber, cork oak, that is Mediterranean. So here you have the distribution area in green. Per species, we have approximately 8 populations. These are the points here, and the warmer the color of the points, the warmer the population. Per population, we have approximately 8 mother tree, and per mother tree, we have 15 acorns. Here I will focus on sessile and pubescent oak, as we already heard about cork oak in Marta's presentation. So what's the population climate for oaks here? The population climate is covering 3 periods. The period from 1900 to 1960, it's a proxy of population local adaptation. The climate from 1960 to 2023, which is a proxy of maternal effect. It's the climate in which the mother tree had developed. And the climate of 2024, which is a proxy of acorn development effect because it's the acorn maturation year. So for the model fit, we need the climate of all our populations for the 3 periods. And for prediction, we are predicting on the whole range. So we need range-wide rasters. Here, for example, I put the rasters for spring temperatures between 1900 and 1960. And we retrieved all these variables, so temperature variables, water variables, seasonality variables, etc., from the Climate.tt website. To test for our plasticity, we use a temperature transfer distance. We conducted experiments under controlled conditions in climatic chambers, where we saw all our seeds from all our populations from the 3 species at 3 temperatures, 15 degrees, 20 degrees, and 25 degrees. And the transfer distance is the chamber temperature minus the temperature of origin, so the spring temperature between 1900 and 1960. And to get our trait data, we measure some traits before sowing and then in the climatic chambers. So we measure seed traits here in brown, seed mass, moisture content. We measure germination traits in green, germination time and percentage. And we measure seedling traits in blue. So we measure the time to first leaf emergence, the height at first leaf, and then 1 month after first leaf emergence, we measure Again, the height to have the vertical growth. We measure the diameter, the root and shoot biomass, the specific leaf area, and the leaf pigments. So now that we have all these data, all these variables, we need to select them to build the model. We use multiple variable selection methods to select the variables. Won't go into details. They are listed here. Basically, we keep the variables that are selected the highest number of times and that have a high rank in random forest and Barita methods. And then we test them via ANOVA and AIC models comparisons until the final model. And then when we have the model, we have to evaluate it, to evaluate how good it is. And to evaluate it, we use some basic metrics like AIC, like R squared and residuals. And as we are making prediction, it's also very important to evaluate the generalizability of the model. So for this, we're doing cross-validation. We use the mean Pearson's R coefficient to evaluate the correlation between observations and predictions, with 80% of data for calibration and prediction and 20% for validation. So how do we know if we have a good model? Well, then when the evaluation metrics are okay, but not only, it's very important that the model is fitted on enough broad and representative range of environmental conditions. And it's only when these conditions are met that the relationships established can be projected onto any climate scenario. Do we have an enough broad range of conditions for our oaks? For the temperature tested, so the climate transfer distance, yes, we have a very broad range, especially towards warming. We have a transfer distance up to plus 15 degrees. 20 and 25 degrees are very warm for some of our oak populations. And are the population representative of the climate covering the whole distribution range? It depends on the species. So here you have the population location within each oak's climatic envelopes. The envelopes is function of the temperature seasonality, the thermicity, which is a temperature index, and the climatic moisture deficit, which is a water variable. And you can see, so on the left, you have a sessile oak, and on the right, you have pubescent oak. And you can see that for sessile oak, it's quite okay. The population are quite well distributed within the envelope. For pubescent, it's not really like this. You can see we particularly miss the driest population. So that means the pubescent models may be less reliable. We have to keep that in mind. So I will now show you some spatial predictions on some traits. These are still ongoing work. The maps are still subject to change, but they make very good example of what we can get. So first, you can see projection for germination percentage. Here it's basically the difference of germination between 1930 and 2080, the end of this century. So on the left, you have sessile oak, and on the right, you have pubescent oak. You can see the scale on the extreme right that goes from minus 15% of germination to plus 10% of germination. Above each map, you can see the model for each species. For example, you can see that the germination percentage in sessile oak depends on the isothermality of acorn maturation year and on precipitation seasonality of origin. It depends also on the temperature transfer distance in orange, which means it's plastic. And for pubescent oak, you can see it depends on the temperature transfer distance too, but with an optimum. It depends also on the spring temperature and the precipitation seasonality of origin. And here there is an interaction, which means the plasticity also depends here on the spring temperatures. And I've put also the R Pearson coefficient, so you can have a evaluation metrics. Here it's almost 80%, which is really good. So what we can see that for sessile oak in particular, the germination percentage in 2080 remained the same or decrease. And for pubescent oak, it's a little bit more contrasted. Here you can see the projection for a phenological trait, which is first leaf emergence time. So it's exactly the same patterns with Petria on the left, pubescent on the right, etc. And here you can see that first leaf time depends on a trait also, not only on the climate. It's in gray and it depends on acorn mass. What we see here is that overall warming really decrease first leaf emergence time, sometimes up to 120 days in some areas, which is expected because temperature accelerate physiological processes, especially phenological ones. Here are relative height growth projections. Likewise, you can see that relative height growth depends also on acorn mass in addition to climate and also on first leaf time for both species. And here overall, we can see that the growth is decreasing with warming or remain the same, and it's especially decreasing for sessile oak. But this is too nuanced because as you can see, the R Pearson is quite low. It's quite weak. It's 0.40. So here are the projections for the total biomass. So the sum of root biomass, stem biomass, and leaves. So you can see the R Pearson here, it's higher than for growth, but it's not very, very high. So this model has still to be checked too. Especially because you can also see that pubescent here is performing worse than sessile, which is not really expected. That does not mean that it's false, but we have to check that it is reliable. And anyway, also talking about the differences between the species, it's not the first time that we are seeing this pattern. And one thing very interesting and surprising that we have seen doing these models is that pubescent oak is often performing less great than expected, especially compared to sessile oak. Now, what's to take home? So model composition is very interesting on the ecological and evolutionary point of view to see on which variable trait depend, if it depends more on temperatures, if it depends more on seasonality, etc. The projections taking into account all populations, that are the ones that I've just presented to you, are great to see the evolution of a trait range-wide with no human intervention. But we can also imagine making prediction using the model fit only on one population to test assisted migration scenarios. And what is really important, how do we improve models and predictions reliability? Well, it's very important to have enough broad range of environmental conditions for populations and environments experimentally tested. Well, thank you very much for your attention. Many thanks to my colleagues and to the projects and organisms that have funded this work. Thank you.